Hi, this is Raj and I'm a PhD student of North Dakota State University. So my advisor is Dr. Quadir and this is the CPM department, and we are in this lab is Research Park 1 Lab 232. So we are mostly working on polymer synthesis and its application in biomedical and different kind of applications. So I want to show you some lab tour of this lab, what we are doing. So this is one equipment that is we are using, that is called centrifuge. So we are using the centrifuge heavily. So this is the high speed and this is moderate speed centrifuge tube, and this is the wood sections we actually use for our chemical synthesis and everything. And for chemical synthesis you always have to take care of. We have a glass door, and inside we have several hot plates. We are using these hot plates for our reactions to elevate the temperature, as well as we can use some magnetic stirring to modify the speed of the solutions, like uniformity. And we also have a pump. This pump is actually is called like syringe pump. So actually we are preparing different kind of polymer beads or something from this syringe. Like you can see over here, this kind of beads we are using, and we are actually loading some drug and applying for biomedical applications. Besides that, we are also preparing some nanoparticle synthesis. Those nanoparticles actually are loading with some drug and other targeted any kind of material, which will do some applications for our own. And we have some we have some nitrogen pumps over here. So you can adjust the nitrogen flow if you need any any reaction, you need any nitrogen conditions, you can use this. These are the bench top, so we are using for our synthesis as well. So this bench top, for each person has their personal bench top, and they are seeing most of the chemical work over here. But if the chemical work would be a little bit harmful or something, then you have to do in a good sections. So this is the sections we actually sit and do the analysis of our experiments. So this is another hood. So this hood, actually we have a evaporator. So what is this evaporator do? Actually it will evaporate whatever solvent you are using for your experiments. And this side also we have our bench top. So each bench top has their pipeters and other all instruments over here. We have sink over here. We do not throw any chemicals in the sink, and we also have a cell biology specified compartment where we only do the cell biology over here. And this is you have to, when you are doing cell culture stuff, you only have to throw the material over here, not in the other portion of this lab. And we also have an incubator which we are using our cells to grow over here. We have two different kind of freezer. So one is 40 degrees Celsius and another is minus 30 degrees Celsius, which we are using for storing our chemicals. We have balance over here to measure the material for your experiments. We have UV spectroscopy measurement instrument, which we are using for any kind of detection of drug or any kind of molecule you want to detect. We have a pH meter. Using the pH meter, we measure the pH level of your material. We have a bath sonicator, which we are using to disperse our material very well in the solutions. And also this is the a chemical situation that we are doing. We throw all our chemicals in a bucket or pail. So actually the liquid waste will go to this and the solid waste will go to this. We are not throwing any kind of any sink. And we also have a first aid kit like if you have any issue, that we have all like essential things over here. We have a documents over here, so you can use to check which chemical you are exposed, and you can treat depending on that. So so we have also HPLC instrument. So this instrument, what will do, like it will detect some drug in a very small scale to help you to measure, and also we have a plate reader to check if you want to measure any kind of analysis, like any kind of like protein, how much protein is there, so you can measure using this plate reader. Yeah, thank you.